Hello guys, welcome to Bosons Botany. In this video, I will define amino acid. So before I start with the structural details and some key points regarding amino acids, we all know amino acids are biomolecules or macromolecules that are the building block of polypeptide chain or protein. And how many amino acids are there? In general, we know that we have 20 amino acids, but actually there are 500 or more than 500 amino acids. But only 20 to 22 amino acids are essentially required by our cells, by the by our body to show different functionalities, to show different metabolic reactions, enzymatic pathways, signaling pathways. So here I will define those 20 important amino acids and two exceptions which are there. So making up 22 amino acid in general. So 20 important amino acids which are very common and known to us, and two exceptions which are known to show anomalous behavior or different. They are slightly different from other 20 amino acids, and therefore they are exceptions. So let us begin with non-polar aliphatic amino acid. We have glycine, alanine, proline, valine, leucine, isoleucine and methionine. These are non-polar in nature. So let us begin with glycine first. So before I start with any of these amino acid, it is important that we understand the skeleton of amino acid. This is the central carbon, and as we know, carbon has 4 valency. It can form 4 bonds, and if a carbon atom has functional groups attached to it, for example, in amino acid we have COOH as one of the functional groups, which is the carboxylic acid group, and NH3 plus, which is the second functional group, that is the ammonium group. We have the R here. R is actually variable. It will define the nature and property structure of particular amino acid. And we have hydrogen atom. This is the simplest structure one can imagine about amino acid. This central carbon is known as the alpha carbon, and this alpha carbon is having the functional groups attached to it, right? Now this R group is variable, as I have already told you. This is variable in nature, and this is the first point. And second point is, if carbon atom has 4 different bonds attached to it, 4 different bonds attached to it, then that carbon is known as chiral in nature. That carbon is known as chiral in nature. So let me just highlight here. This is group 1 or atom 1. This is atom 2. This is atom 3. And this is atom 4. So we have 4 different atoms bound to this carbon atom. This is the carbon atom. that we need to study, and this has 4 different bonds. So this is known as chiral carbon. I mean this carbon atom, okay, because this has all 4 different bonds. 4 different bonds. But if I make one of the bonds similar to the other bond, like if I erase this here and okay, so as you can see, these 2 bonds, this bond and this bond, they are similar. So this carbon is no more chiral in nature. So this is now known as achiral. Achiral carbon, because here now 4 bonds are not different. 4 bonds are not different. So I am cutting this different word here. So chirality is basically defining how many different bonds are there to a carbon atom, and if a carbon atom is chiral in nature or asymmetric in nature, then that carbon atom will be chiral, and it will rotate the plane polarized light in either dextro or levo direction. Dextro means towards the right side and levo means towards the left side. And if a carbon atom has similar 2 bonds or 3 bonds, not all the 4 bonds are different, but 2 bonds are same, 3 bonds are same, so that carbon is known as achiral in nature, and that will not rotate the plane polarized light. So these some are very essential points which you need to keep in mind before starting with amino acid structure, because amino acid, we differentiate each and every amino acid based on R group, and whether the alpha carbon or the central carbon is chiral or achiral in nature. So if you understand, let me proceed to the structural definition of amino acid. So let us begin with non-polar amino acid. So we have the most simplest amino acid, which is glycine. Glycine, it is extremely simple. As you can see, the R group here, it is hydrogen atom, and this is the alpha carbon because it has 2 functional groups attached to it. Now this glycine, it is achiral in nature. Why? Because this R group is giving hydrogen atom here, and this in general is having hydrogen. So hydrogen atom of the amino acid and hydrogen atom due to this R group. So glycine, it is achiral. Achiral in nature means it is symmetric. So it will not rotate the plane polarized light, and therefore there are no stereoisomers. And it is the simplest amino acid. Now, how will you calculate stereoisomer? Number of stereoisomer is calculated by using the formula 2 raised to power n, where n is number of chiral carbon. Number of chiral carbon. So this is the simplest formula that you need to remember to calculate number of isomers or stereoisomers. I hope it is clear. Now let me move to glycine. Glycine is the second simplest amino acid because here also the R group is not very bulky or it is not very complex. It is just a methyl group. And the alpha carbon here, it is chiral in nature. Why? Because all the 4 bonds are different. So it will definitely rotate the plane polarized light in either of the directions, such as levo or dextro rotation. And alanine, as you can see, there is only one chiral carbon. There is no other chiral carbon. So how many isomers we will get here? 2 raised to power n, which gives us 2 isomers. Now let's move on to proline. Proline is bulky because it is having a very huge R group. This is the cyclic R group, as you can see, which is making a 5 carbon cycle. So this is the R group, and proline is known as helix breaker. Why? Because it will disrupt the secondary structure, which is the alpha helix, if it is present in between 2 amino acid. For example, if amino acid 1, then proline, then amino acid 2, these 2 amino acids were required to form alpha helix, but proline is present in between these 2 amino acids, so the alpha helix will be disrupted, it will be destabilized. So proline will not allow alpha helix formation, and this is also chiral. This alpha carbon is chiral in nature. So let me tell you one thing. Out of 20 amino acid, only glycine amino acid is the achiral amino acid. It is symmetric, or it will not rotate the plane polarized light because it is not having a single chiral carbon. Rest all are chiral in nature. So here also we have 2 isomers. 2 to the power n means 2 isomers. Now valine. Okay, before I start with valine, in non-polar aliphatic amino acid, we have 3 branched chain amino acid. 3 branched chain amino acid, which are valine, isoleucine, and leucine. These are 3 branched chain amino acid. And what is the meaning of aliphatic? Aliphatic means there is no aromatic chain, or no benzene ring, or no aromatic chain. Okay, that will fulfill the Huckel's rule to define whether it is aromatic or non-aromatic. So there is no aromatic group attached to the R group. R group means this highlighted part. Okay, so now valine is the branched chain amino acid. As you can see, this is the branched chain amino acid. And how many chiral carbon is there? Only one. This Why not this one? Because definitely there are two similar bonds, CH3, CH3. So this carbon is not chiral in nature. So again we have 2 raised to power 1, which is 2. Now after valine we have leucine. We have leucine here. Leucine, this is again a branched chain amino acid, and again we have only one chiral carbon, this. So 2 raised to power 1, which is 2. Why not these and these? Because this carbon has 2 hydrogens, so same bonds. This carbon has 2 methyl groups, so same bonds. So only one chiral carbon, right? We have isoleucine. So isoleucine, this is again branched in nature, but one key point which you need to remember, we have 2 chiral carbon here. First, this alpha carbon, this is chiral, and this carbon beta, this is alpha, this is beta, this is we nomenclate carbon atoms like this, alpha, beta. So this beta carbon, this is chiral in nature. So let me highlight these 2 chiral carbons, this and this. These are 2 chiral carbons we have. These are 2 chiral carbons. Why? Because chiral carbon 1 or chiral carbon alpha, this has first bond, second bond, third bond, and fourth bond. This fourth bond means this whole R group. This R group. These 4 bonds are different. So alpha carbon is chiral in nature. And beta carbon, this is also chiral in nature. Why? Because this bond, and this bond means having all these atoms. Okay, this complete group here, which is bound to carbon beta. So bond first, bond second, bond third, and bond fourth. Bond fourth means this whole part. So all the 4 bonds here in case of beta carbon are different. So carbon alpha and carbon beta, these 2 are chiral in nature because their bonds are different, or all the 4 bonds are different. So let me write here, isoleucine has 2 chiral carbons. Therefore 2 raised to power 2, which is 4 isomers. So isoleucine is important in terms of chirality. Then we have methionine. Methionine is the sulfur containing amino acid. As you can see, there is sulfur in between these 2 methyl group, and this sulfur is non-ionizable. This cannot be ionized because this sulfur is in between, not towards the end or towards the terminal. So methionine is known to have a sulfur group, right? And also there is only one carbon which is chiral in nature. So 2 raised to power 1, which is equals to 2. So these were the aliphatic non-polar amino acid. I hope it is clear. Now let us move to aromatic R groups. Very simple, and only 3 in existence. These are Phenylalanine Tyrosine and tryptophan. Why they are aromatic? Because they have aromatic chain or benzene ring. Here we have benzene ring, so we have benzene ring attached. and benzene is aromatic in nature, as we all know. So let us start with phenylalanine. By the name itself, we have phenyl, means this benzene ring, as you can see, and this is the R group. Then tyrosine, we have OH bond, OH group attached to this phenyl group, benzene plus OH phenol. So this whole structure is the R group. And tryptophan, we have this indole ring. This is known as the indole ring. Indole ring. And this is the benzene ring here. So there are 3 aromatic amino acid: phenylalanine, tyrosine and tryptophan. Now, one thing that I need to mention, non-polar and aromatic amino acids, out of these, how many are polar? I mean, how many can be polarized when we dissolve these amino acid in any medium, any chemical medium? Then what are the groups which can be polarized? So let me just mention them here, because during charge calculation we need to remember polar groups or polar amino acids. So in case of non-polar amino acid, by the name itself, these all are non-polar. So no polar group we have. All are non-polar. All are non-polar. Okay. In case of aromatic amino acid, we have tyrosine as our polar amino acid. Why? Because here we have OH. And if I explain this part here, this OH can be written as OH like this, and oxygen is highly electronegative in nature, so it has lone pair 1, 2, 3, 4 and 5, 6. 1, 2, 3, 4, 5, 6 like this. These are lone pair of electrons. So if we dissolve tyrosine in any chemical medium, then this OH group or this OH bond will have the tendency to cleave and release hydrogen ion like this. So hydrogen ion can be released into the medium. Why? Because we have this highly electronegative atom, which is oxygen. So in general, OH group gives polarity to tyrosine. So tyrosine is polar in nature. Tyrosine is polar in nature. Tryptophan, it is non-polar. As you can see, there is no ionizable group. And one more thing, if we exclude R group, every amino acid has 2 common polar groups which are always ionizable and which will be always polar in nature, and these are our functional group, that is this COO minus and NH3 plus. These are polar in nature, but we judge the type of R group which is present or bound to carbon alpha. If R group is non-polar, then we will say that the amino acid is non-polar, and if it is polar, then the amino acid is polar in nature. I hope this point is clear. Now let's move on to polar uncharged R group. So polar uncharged R group, we have serine, threonine, cysteine asparagine and glutamine. Uncharged because R group is having no charge, positive or negative, no charge. So serine, as you can see, we have the OH group here which can be polarized, so it is polar in nature. Threonine, this is having OH here, so it can be polarized. And another point, there are two carbon which are chiral in nature. First, this alpha carbon and this beta carbon. Why? Because alpha carbon has first bond, second bond, third bond, fourth bond, all different. And this beta carbon, it has first bond, second bond, third bond and fourth bond different. So here also 2 raised to power 2, which is equals to 4. So isoleucine and threonine, they are chiral and have 2 chiral carbons. Okay, so I hope this is clear. Now cysteine, this is again polar in nature. Why? Because we have this terminal SH group here, terminal SH group having sulfhydryl group. We say it is having sulfhydryl group. And cysteine can make disulfide bond, that I will define in my next video. So in my next video I will emphasize on each of the amino acids. So in this video, not explaining what is disulfide bond, but cysteine is known to make disulfide bond because it has sulfhydryl group here, SH group, which is ionizable. We have asparagine, this is the amide group, this is the amide group, this. And we have glutamine, this is again we have this amide group. So these are polar uncharged R group. Okay, polar uncharged R group. Now we have positively charged R group. Positively charged means they are basic in nature, basic amino acid. Why basic? Why negative? In my future video, where I will define charge calculation and pi values Zwitter ion, there I will discuss about basic and negative, basic and acidic amino acid and why they are known as positively charged and negatively charged amino acid. But for now, just remember that positively charged amino acids, they are basic in nature. So this is lysine. As you can see, there is one extra NH3 group that is giving its basic character, and this is having positive charge over here, so it is positively charged. This is arginine, which is having this NH2 group here and another NH2 group here, so 2 NH2 group we have. This is histidine, which is cyclic in nature, having this NH group here and another N here. Okay, so these are positively charged R group and basic in nature. Now last but not the least, negatively charged or acidic amino acid. Acidic amino acid. Why? Because we have second carboxylic group attached here. So these are acidic in nature, aspartate and glutamate. Now if we hydrolyze glutamine, then we will get glutamate, and if we hydrolyze asparagine, we get aspartate. So this is very important. Asparagine, ASN, 3-letter code. Asparagine, when it is hydrolyzed, we get aspartate or aspartic acid, and glutamine, GLN, when it is hydrolyzed, we get glutamate. And in my next video I will discuss about 1-letter and 3-letter code, then the key points or important points regarding all these 20 amino acids that I have described here. So it is very important to remember all these structures, at least R group, what are the characters these R group have. So let me wind up by writing what are the polar, non-polar aromatic amino acid. So non-polar amino acid, we have glycine, alanine, valine, leucine, isoleucine, methionine. Then we have yes, that's all I guess. Leucine, isoleucine, methionine, valine. Okay, we have proline. These are our non-polar amino acid. Polar amino acid, we have tyrosine, serine, then we have threonine, then we have tyrosine, serine, threonine. What else we have? Let me check. Threonine we have, cysteine we have, threonine, cysteine. Then cysteine we have. Then acidic amino acid we have aspartic acid, glutamic acid. Basic amino acid, lysine and arginine. Then aromatic amino acid, we have tryptophan, tyrosine, phenylalanine. So I have written all the single letter code abbreviations. So before I end this video, let me just write down the single letter code here adjacent to these amino acids so that you can note them down. For glycine it is G, for alanine it is A, for proline it is P, for leucine it is L, for isoleucine it is I, for methionine we have M, then for serine we have S, for threonine we have T, cysteine C, then asparagine we have ASN as the 3-letter code, and for glutamine we have GLN as the 3-letter code, and for glutamine the single letter code is Q, and for asparagine it is G, no, not G. Okay, I'm not remembering the single letter code for now. In my next video I will tell the single letter code of asparagine. Okay, I have just written the 3-letter code here. Now for aspartate or aspartic acid we have D, for glutamate we have E, then for histidine we have H, for arginine we have R, for lysine we have K, and for aromatic amino acid here phenylalanine is F, tyrosine is Y, and tryptophan is tryptophan is W. So I hope this video makes some sense and you get an overview of amino acid. So in my next video, I will describe each and every amino acid and some key points which you need to remember. Thank you.